Hey, it is my pleasure today to introduce Dr. Stan Floresco, who's a professor of psychology at the University of British Columbia. Hey, Stan.
Hey, Dr. Muller, how you doing?
Very well. So Stan is a person who's had a long relationship with the journal Psychopharmacology, in that he's published, I think it's 14 papers in the journal over the years. So it's been kind of a key journal for his publishing in general for his lab and as a trainee. And he's also an editor, a principal editor for the Americas in preclinical psychopharmacology and translational studies. So Dr. Floresco, could you tell us a little bit more about your sort of history with the journal Psychopharmacology?
So when I first got into this line of work in the mid 90s, last century, and I first started kind of reading original research papers based on work I was doing in the lab as an undergrad on nucleus accumbens and the dopamine. These were some of the papers that kept coming up in terms of where I was doing my research, trying to find new information. And so, in my opinion, it's always been just like a fundamental behavioral neuroscience journal. I mean, based on its entire history, when you look at it, some of the kind of monumental findings based on certain assays, like reinstatement paradigms, forced swim, things that are kind of commonly used. A lot of them are published in psychopharmacology. So I've always viewed as a foundational journal. And then as I moved as a trainee and then as a PI, I've always kind of looked to this journal to publish work where we really wanted to kind of describe in detail, the behavioral effects of drugs and really kind of pick them apart. I think in this line of work in particular, in behavioral neuroscience, one thing that those of us trained in that field do when we're doing it properly is we're not just telling you what a drug or certain brain manipulation does. We also kind of do our proper controls and say what it doesn't do to help kind of refine our understanding of that. And I've always found psychopharmacology has been kind of a great journal for publishing that type of work. I found the review process to be vigorous and, keeping us honest in terms of, what is the alternative interpretation for some of the behavioral effects you're seeing. Students and junior investigators also trying to figure out where should I publish my work. You have a data set, where to go. And often I say, if you don't have a clear target, look at your reference list and see, the papers you're citing. What journals are they in? Because that's what you really want to do, is send something where an audience is going, your audience is going to see that. And it just so happens for a lot of the stuff that we do, we look at our reference list and there are a lot of papers we're citing from psychopharmacology. And so that's another kind of reason why the work that we've published there has been sent to that journal.
Interesting perspective. So now as an editor, you're sort of not just deciding where to send your own papers, but you're also deciding, is this paper, a good fit for this particular journal, let's say psychopharmacology. Can you tell us a little bit more about how you make those kinds of decisions for psychopharmacology, or maybe in comparison to other journals that you review for, edit for?
Yeah, so I because, it's a drug journal, there are certain things in pharmacology, in the field of pharmacology research in general, that, we have to pay a little attention to detail. And in addition to kind of general things about controls, procedures, interpretation, statistics. So I'm always kind of looking for, are you doing an appropriate dose response curve when you're making a claim? Studies that just, use a single dose of one drug, particularly if it's a novel compound or relatively untested, if it's not well established, that's something that I'll, always kind of be wary of in terms of kind of making my decisions and at least suggesting to the authors, you kind of need to do more of that. Also, one thing I see a lot of, and it winds up being a red flag for me, is that you might say have a compound that you're using in combination with some animal model of some psychiatric disease, say some sort of stressor to model depressive-like symptoms, and you want to ameliorate those symptoms on your behavioral readout with said compound. And they'll do kind of, the stress or your manipulation versus animals that didn't get that manipulation and see your effect. And then you have another group where you get the manipulation and your drug. But they're missing what I always think is a key control, the drug by itself in intact animals. I think it's always important to see, especially if you're trying to develop something that might be a potential therapeutic. I really think it's our responsibilities as editors to kind of refine what gets out there and make sure, okay, we have a good understanding of what the basic function of this compound may or may not be, and otherwise normal states, before we can kind of make any strong assumptions of what it's doing in perturbed states. And in terms of kind of, sometimes you'll get a reviewer asking for more experiments, and we can always do more experiments. And part of it, I've always taken the point is like, when is it time to just to get to your endpoint? When do you have enough of a story to publish it? And so I personally, I can't say all editors do this, but I'm kind of wary to propose that an author does new experiments on a revision, unless it's kind of a crucial thing based on their interpretation of the data. So if they are saying, this is what we think this effect means, but they haven't ruled out an obvious alternative interpretation with a key control experiment, well, that's when it's like, okay, if you want to say this, you need to do this additional experiment. And so that's kind of a policy I've always taken, because I'm an author too, and I don't always like having to do more work when I think I have a complete story. But one thing actually that surprises me is often I will give authors a way out, and I'll say, ideally, this would be the best thing for you to do this additional experiment, or at the very least, can you cite other data, give more rationale as to why you think this is not an issue? And more often than not, authors will actually do that experiment. I think they're finding the exercise useful. They want to know themselves and be absolutely sure that their interpretation is a little more airtight. So, and that's something I always kind of find, like, I admire about scientists. Is like, I'm giving you an easy way out. No, we'll take the harder road to be absolutely sure what's going on.
Really interesting perspective. I wonder, it just sort of occurs to me that, this idea of adding new experiments when there's a, sort of key interpretation at stake, has that been something, or I guess maybe even more broadly, has the COVID pandemic that we've gone through, and you being an editor in that period, obviously a lot of things changed in our lives. And I'm just curious if you can say anything about how that impacted your role as an editor for a scientific journal, if it did.
It's so, first thing I can tell you, because both for Psychopharm and for other journals that I work for, submission rates went up a lot in the first year of COVID, because everybody was working from home and they all had backlog data and they were submitting it. I did the same myself. And then the next year, submissions dropped a bit, probably because they hadn't collected data for the last year. They were just writing it up. And I think that when we were getting requests from the reviewers for a particular group to do additional experiments, that factored in a bit more. And I was even a little bit more hesitant, although it didn't wind up, for a lot of the stuff that I handled, didn't wind up being a major issue, but I was always kind of wary of that. And if there was a situation, I'd always try to kind of let the authors know, we understand it's a weird time to be doing this right now. So if there's another way you think you might address the reviewers' concerns, we'll do that. And I also think, from my perspective, and I think a lot of other editors, when you're dealing with comments, reviewer comments, realize that it's ultimately the editor that makes the decision. The reviewers advise, the editors decide. And so keeping that in mind, you don't just need to convince the reviewers, you can convince the editors first, and that kind of will ease your way through if the paper is going to make its way into print.
Wow, yeah, it seems that that's like sort of a important thing to consider when composing revisions to a paper after it's been through the editorial process. If you have an antagonistic reviewer, your job is not necessarily to do every nitpicky thing they're telling you to do, but actually to just appeal to the editor, essentially, to say, like, these are nitpicky, stupid little things, and this is a good point, etc. And if you make a strong case in that sort of way, I think usually that's going to go pretty well for you.
Yeah, when I craft my rebuttal letters, I even though sometimes I'm very tempted to use slightly harsher diminutive language, as a Canadian, I try to be as polite as possible. And
it's harsh when you don't say sorry after.
Yeah, well, it really, no. I always apologize to my review, even though it's not my fault. It's like, hey, sorry, I guess you missed it. We didn't type things clearly enough. Maybe our font choice was not the best for you to see these effects and data that we clearly presented in our text. But it just sometimes it's you gnash your teeth a bit when you're typing. We are grateful to the reviewer for pointing this out. But it's I've always been a fan of the adage that if you make it easier on the reviewer, they will make it easier on you. So just kind of be absolutely clear, be thorough. Certainly as a reviewer and as an editor, when people are doing their revisions, I see a lot of, in response to a comment, we did this. No mention of where it might be in the manuscript, no mention of how you did it, just we corrected this. And so that makes it harder on everybody to figure out, did you actually address this? So I think a more detailed rebuttal letter, being clear in your manuscript, highlighting where the changes you made, I find tend to help papers sail through easier. This is particularly the point when it might be the editor making the decision rather than the reviewer. Sometimes you don't secure the reviewer again on a re-review, and the editor has to make the call. And they have to go, they read the manuscript, they go through it. And if I'm doing this and I want to see, did you address the comments the reviewer brought up, and I'm having problems finding it, well, it just makes it harder for me to make a decision, right? So there is a psychology to the whole editorial and review process. And those of us trained in psychology departments, I think, tend to have an inherent advantage of this.
Yeah, fair point. Yeah, one thing I've noticed occasionally is, if an author is very sort of aggressive in their response, meaning like almost like just arguing every single point that the reviewer is bringing up and saying it's not a problem and something like that. Yeah. That doesn't tend to work very well. Similarly, if you're rebutting a particular reviewer point, my philosophy is that should be in the paper too. That's why the reviewer is mentioning it. The rebuttal alone is not, that's not really like public in most journals, including psychopharmacology. So really it should reflect some kind of change in the paper that addresses in some manner the reviewer's point. And on the other flip side of it, you don't want to be acknowledging every single silly little point, doing everything that an unreasonable reviewer is telling you to do. I think there's a way to kind of tell the editor what you think is a reasonable point and what you think is silly, and act accordingly.
I completely agree on both points in terms of being aggressive as an author. I've always kind of viewed when we've gotten critiques on a paper, it's always okay, I clearly wasn't clear enough, or I didn't cover all my bases. If there are unreasonable things, then it's at the very least, let's put something to in the manuscript to acknowledge this issue that was raised. Even if I don't think it's that important, maybe someone else does. Okay, so I'm at the very least acknowledging the issue and saying why it may not be as big a deal in terms of our interpretation results. So yeah, I totally hear you there.
Cool. And then just to bring in conversation back to the topic we were discussing a little bit before, that psychopharmacology is particularly good at sort of pharmacological approaches to this kind of behavioral studies, behavioral neuroscience studies many times. Obviously there's been a lot of advancements over the course of your career, my career, even trainees' careers in methods, especially. And, a lot of times these kind of new methods are, fundamentally different in the way that they manipulate the brain and that kind of thing. And so a lot of people really give a lot of attention to them, even if it's sort of like the behavioral findings, for example, that come out of them are not necessarily totally different than conventional approaches to neuroscience. What can you speak a little bit to like your perspective on this idea of methods as like being attractive for their own sake almost, versus what they specifically tell us about the brain, and how that plays out at a journal like Psychopharm or others?
Yeah, so I mean, obviously, the majority of the work that I handle as an editor is still kind of drug-based, but we are starting to see more and more papers using chemogenetic approaches, so on the occasional optogenetic approaches, often targeting neurochemical pathways, so still falling within the realm of pharmacology. It's just a very novel way of manipulating brain neurochemistry, because that's ultimately what we're talking about. But I mean, I think as we get newer approaches to more selectively target cell types, to get greater temporal specificity in manipulating brain activity to understand behavior, the fact is that drugs in our society, therapeutic and recreational, are still going to be part of our society for a long time. And with all these new approaches that come out, one thing that, one of my former colleagues at University of Pittsburgh, Susan Cisak, said, it's like, the difference between kind of the new methods and the old methods, like, we know all the limitation of the old methods already. They're still effective, and at least we can guard our conclusions based on the known limitations. But you still keep seeing with some of the newer techniques, new papers coming out, well, this is not always doing what you're doing. I mean, obviously, examples with off-target effects of CNO with chemogenetic manipulations, or issues with optogenetic inhibition or stimulation. Are you getting rebound excitatory effects when you're inhibiting these things? And so these are still coming out. And so I still kind of view myself as an old school behavioral pharmacologist. And I think that there is still kind of a lot of validity in that approach to addressing certain questions. There are some questions that you really need these new techniques to address. You can't do it with old school, but there's still a lot of stuff out there in the brain, certain brain regions, certain behavioral functions, neurochemical targets that you can get new insights, both into basic function and potential therapeutics, by using kind of the old school psychopharmacological techniques. And so again, I think this is why our journal at Psychopharmacology really serves a purpose to the field. And I mean, another great thing about the journal that doesn't get highlighted as much is we don't just do kind of basic animal preclinical research. We also have, a good number of human psychopharmacological studies that are published here, and a lot of translational stuff. So again, I think it's a cornerstone for understanding, both the neurochemistry underlying normal and abnormal behavior.
Yeah, well, speaking of that, we've talked a fair amount about the journal's role in promoting pharmacology research of various kinds, or as a key journal for that kind of research. Let's talk about the psycho part. So the relevance of some of the behavioral models that we use in these in a lot of our preclinical neuroscience experiments have been questioned by a lot of people. A lot of these behavioral models were actually first published in psychopharmacology, some of the most sort of famous ones in the history of like kind of depression drugs and perhaps schizophrenia drugs and so on. Can you speak a little bit to your take on how behavioral models of animals should be related to the human condition and to normal and abnormal behavior?
Yeah. So, okay, first, let's just kind of clarify terminology here, difference between models and assays. And so, I always try to use the term models. It's like, okay, I've done something. I've manipulated a biological system in a certain way that I think may approximate a disease state or some other condition. The assays for psychopharmacology in particular, these are our behavioral readouts. So this is what we really care about in terms of using them with our models. And certainly, some of the earlier assays that were developed, like things like reinstatement and other assays that have since been critiqued, they still have provided information to the field. And not just in terms of, okay, they were useful, but even their critiques has provided us information and helped us refine, well, exactly how are we going to use behavior in animals to maybe understand how this might relate to humans, to therapeutics in humans. And there has been, in our circle, Steve, there have certainly been conversation for a number of years of the issues with different behavioral assays. And the concern is that they're not particularly predictive, especially when we're looking at drug discovery efforts, that these assays are not necessarily predictive. And so we can have a relatively rudimentary assay in animals, in rats and mice, and we see an effect with a new compound, and then it fails in phase one. And my opinion is that a lot of these times, it's probably because your assay is not very translatable. You're not measuring the same construct that you might be in the assays you're using with humans, or for other reasons, or the reason you might see, say, an enhancement in cognition or emotionality in animals is your behavioral readout. You saw a change in behavior, but your readout is not a true index of the process you think you're studying. And so I think this has actually been another kind of useful aspect of this journal in particular, is because we continue to kind of publish articles that develop and that describe new procedures, new behavioral procedures that other people wind up using and then can kind of take the field forward. And I think one issue is honestly just getting funding to develop these assays, because the funding agencies will say, okay, well, your assays are very rudimentary. You need to develop some, but they won't give you money to actually do the hardcore legwork to really validate tests. As someone who's developed new tests measuring decision making and whatever, it takes a while, and often a lot of incarnations of changing your procedures that can take 2 to 3 years just to kind of get your basic assay that you're confident enough might be measuring something you think you're measuring. And so I think our journal kind of always welcomes those types of studies that have really kind of done the legwork and developed new assays. And I mean, as it turns out, a lot of those papers wind up getting cited a lot. Maybe not within the 2-year window of the impact factor, but lifetime citations of those types of papers tend to get cited a lot. Once people realize, hey, this is a cool way of measuring this.
Yeah, and some of the most cited papers in psychopharmacology are exactly that, developing assays for various psychiatric disorders, for better or for worse, perhaps we could say.
Yeah, true that.
But
you
yeah,
always got to start somewhere, and you can't fault some of the older assays because that's all we had at the time. And I mean, some of them developed in the 70s and 80s before we had all these additional resources of information and citations and communications with other groups and new ways of analyzing behavior. So it's I think the concern is that some researchers in the field who are very sophisticated with their kind of neural manipulation techniques or molecular manipulations, genetic, what have you, are taking old school, just boilerplate assays as a kind of just and not necessarily thinking about how we might really test these effects. And I mean, everyone's got their own expertise, and someone who's got a lot of molecular expertise really doesn't have the background to develop a new behavioral assay. And so I think people like us still serve a really important function in this field. And as I said, this journal serves an important venue for people like us to talk about that.
Yeah, and I just add that this is, in terms of, identifying that we've had an issue in neuroscience with, defining depression based on forced swim, for example, because that's
glad you said it first.
Drugs and so on. That's just one specific example, but that's how this is the scientific process. Is we have a hypothesis. There's a logical reason to think the behavior on forced swim is related to depression based on some conceptual ideas, and the drugs that we've developed based on that haven't really worked in the way that we wanted them to, and that's how we make progress. Though we correct things that we didn't do as well before. That is science.
Yeah, absolutely. I think also, I mean, the field is really benefiting from this RDoC approach that are using now, and rather than clustering things into disorders, let's look at symptom clusters that may be common across disorders. And I think, again, preclinical researchers really benefit from it, because this is what we've been doing for years. We've been looking at specific constructs, and it benefits not just in terms of us being able to study in animals, but now we can develop assays that are sometimes easy to translate across species. So, I mean, one of my kind of favorite examples related to the work that we do is looking at kind of effort-related decisions and motivations. A lot of this foundational work was done by John Salomon, and a lot of his stuff was published in psychopharmacology. And he was just doing this work for many years, initially as a means to an end for trying to identify what dopamine does and doesn't do. Because as we both know, the old law was dopamine was reward and pleasure. And of course, I still get antsy just saying that, because we don't necessarily believe that. And trying to kind of refine what it actually might be rather than a hedonic signal. And then it got picked up as like, hey, these effort-related choice paradigms, they're actually a form of decision-making. And then human researchers started getting into that. It's like, hey, you know what? There are motivational deficits in things like depression. Let's develop an assay based on the rodent assays that can test that. And lo and behold, individuals with depression show these types of impairments that you see when we do manipulations that are relevant to depression, not just pharmacological manipulations, but stress or targeting certain hormone pathways. So I think that is a really nice continuum that we're seeing when we're getting more and more human researchers kind of looking at the old animal literature and saying, can we develop a task that might mimic something that's preclinical? Because the old days was always like, okay, we have a primate or a human task, can we get rats to do it? Now we're trying to do it the other way, and I think that's really moving the field forward.
Absolutely, yeah. I think the future is both of these things interweaving together as much as possible, so we can get as much information about what is and is not similar in animals and humans.
Yep.
Great. Well, I guess with that, we'll wrap up, and I'll just thank you again, Dr. Floresco, for your time and this really interesting conversation about the journal Psychopharmacology and your career, how your career is intertwined with it.
This was a lot of fun. I really appreciate you inviting me.
My pleasure entirely.